Hello, so today we'll discuss about features or characteristics of DNA. So first it is made up of two polynucleotide chains and here the backbone consists of sugar and phosphate, and bases they project inside, like they are towards the inner side. Then they means like two chains. There are two chains. One, they have anti-parallel polarity. Anti-parallel means one is 5' to 3', other is 3' to 5'. Here you have to look for the arrows. Sometimes it's like 5' to 3', or sometimes it's written like 3', 5'. So we have to look for the arrows. This comes in sense strand and in anti-sense strand. Okay, then next sometimes what happens, the question comes like they will not give you numbers. Okay, so they will give you like, here we have phosphate, here we have OH, what will be the direction. So you should know that 5' is phosphate and 3' is OH, so it will be 5' to 3'. Sometimes it's written 5 phosphate is here, OH is here. You have to make out which direction will be this. They will not give you numbers. You should know 5' is phosphate and 3' is OH, so it will be 5 to 3 like this. And then third is hydrogen bonds. The bases in two strands, they are paired through hydrogen bonds. Bases like if I A double bond T and C triple bond G. So here two hydrogen bonds are there, and here 3 hydrogen bonds are there. So purine comes opposite to a pyrimidine. So purine is adenine and guanine. This is purine, adenine and guanine. You might have seen purine is always opposite to pyrimidine. Okay, then next, right-handed fashion. The two chains are coiled in right-handed fashion, and coming to pitch of helix, it's like if I say this is a DNA, okay, the pitch of helix, that it is 3.4 nanometer. So this is 3.4 nanometer. Next, nanometer means 10 raised to power minus 9 meter. So there are roughly 10 base pairs in each turn. And if I talk about distance between base pairs, distance between base pairs, that is 0.34 nanometer. Distance is like this. If this is the DNA, the distance will be like this. Okay, then plane like stacking. Next you can stacking, next point. So the plane of one base pair, stacks over the other, like second base pair stacks over the plane of second base pair. So this what happens, this provides stability to the helical structure. This gives stability to the helical structure. Now how this is, let's say if I draw DNA structure like this, here we have A double bond T, here we have C triple bond G. So this is one base pair, this is stacking, like, or you can say this is lying over the other base pair. So all this is done with the help of hydrogen bonds. So this gives us stability. So this is like 3.4 and this is 0.34. Units are most important if they are talking in, we are talking in nanometers. That means 10 raised to the minus 9 meters. You have to see whether it's given in nanometers or in meters. If you want to convert it in, if you want to convert nanometer into meter, you just simply write it like 3.4 into 10 raised to the minus 9 meters or 0.34 into 10 raised to the minus 9 meters. Okay, then there is a, then coming to next we have central dogma. So then Francis Crick proposed central dogma model. Or like usually we say Crick. So here he told about the genetic flow of information. Now how this is done? This is done by DNA forms mRNA. Now how DNA is formed? This is the process of formation of DNA, that is called replication. Then how RNA is formed? RNA is formed from DNA and that process is called transcription. Then from mRNA proteins are formed and the process of formation of proteins from RNA is called, that process is called translation. So this is central dogma. If I say reverse dogma, you will just reverse it, protein to RNA, RNA to DNA. For example, in viruses, RNA to DNA, this is the process in viruses. So this is basically reverse central dogma. This is just extra part, you don't need to remember. Okay dear, thank you.